Welcome to the Creative Biolabs Science Channel. Today, we're diving into the world of virus-neutralizing antibodies, exploring how they function and discussing applications of these antibodies. In this video, we will introduce what antibodies and neutralizing antibodies are, the production and regulation of neutralizing antibodies, their mechanisms of action, classification and applications, as well as the challenges they face. First, let's break down the fundamental idea of antibodies. An antibody, often referred to as an immunoglobulin, is a Y-shaped protein that's mainly produced by plasma cells, which are a kind of white blood cell. These antibodies are crucial elements of the immune system. They are specifically designed to identify and attach to antigens found on the surfaces of pathogens. This attachment plays a vital role in neutralizing the pathogen by either preventing it from infecting cells or tagging it for elimination by other immune cells. Antibodies are like the body's own security team, each with a specific target in mind. Here's how they work. First, they spot and latch onto unique markers, called antigens, on pathogens. Once attached, they block these baddies from invading our cells or wreaking havoc. They also flag pathogens for cleanup, making them easier for immune cells to eat up. Plus, they kickstart a reaction that can obliterate these invaders. And when things get really serious, they call in backup, like natural killer cells, to take out infected or altered cells. All these actions are crucial for keeping us healthy and fighting off infections. Neutralizing antibodies are a special kind of antibody that protect against pathogens, especially viruses, by attaching to them and preventing their entry into host cells. They do this by targeting and disrupting key structures or processes the pathogen needs to infect and replicate within cells. By blocking the pathogen's access to cells, neutralizing antibodies halt the infection and prevent the virus from spreading within the host. These antibodies play a critical role in safeguarding the body. After a viral infection, the body produces antibodies, including both neutralizing and non-neutralizing antibodies. Only neutralizing antibodies can deactivate the virus, allowing it to be engulfed by cells. While non-neutralizing antibodies also bind to pathogens, they do not block infection directly. Instead, they might tag the pathogen for destruction by other immune cells, facilitate the removal of the pathogen through phagocytosis, or trigger other immune responses that contribute to the pathogen's clearance from the body. Non-neutralizing antibodies may bind to parts of the pathogen that are not directly involved in the process of infecting host cells, or they may bind in a way that does not interfere with the pathogen's ability to enter cells. When a virus invades the body, it's immediately targeted by our immune system. Antigen-presenting cells, such as dendritic cells and macrophages, capture the virus and showcase its components on their surfaces using MHC class II molecules. This display prompts helper T cells to recognize these viral fragments, triggering their full activation with additional signals from the presenting cells. These activated T cells then bolster B cells that have processed the same virus, helping some transform into plasma cells. These plasma cells become antibody factories, producing and secreting antibodies tailored to fight the specific virus, releasing them into the bloodstream to neutralize the threat. The production and effectiveness of neutralizing antibodies hinge on multiple regulatory elements within the immune system, ensuring a precise and potent response. Key players include helper T cells, which not only kickstart B cell activation but also release cytokines to steer their evolution into plasma cells, the main factories of antibodies. The quality of antigen presentation by cells like dendritic cells also plays a crucial role, initiating T cell activation, which is pivotal for B cell activity. Additionally, regulatory T cells and immune checkpoint proteins like CTLA-4 and PD-1 help moderate the immune response, preventing overactivation that could lead to autoimmunity. Furthermore, the strength of the bond between B cell receptors and antigens dictates the effectiveness of the resulting antibody response, while some microbes may dodge these defenses by altering their surface markers, challenging antibody recognition. Neutralizing antibodies are specialized immune defenders, crucial in battling viral infections. They operate by first recognizing and attaching to specific antigens on a virus, usually the proteins it uses to bind and enter host cells. Once attached, these antibodies block the virus from connecting to and entering the cells, preventing it from taking over the cell's machinery to replicate. This blockade stops the virus right in its tracks, halting its spread within the host. But that's not all. These antibodies also tag the viruses for destruction. Bound viruses attract immune cells like phagocytes, which engulf and destroy them. Furthermore, neutralizing antibodies can kickstart other immune responses, including activating the complement system. This part of the immune system attacks and breaks down the virus's cell membrane, neutralizing it completely. Let's delve deeper into the working principles of virus-neutralizing antibodies in vitro. Researchers have identified multiple mechanisms of action for antiviral neutralizing antibodies. One primary method is preventing virus particles from attaching to host cell receptors. This is achieved by disassembling the viral spike proteins or altering their conformation. Additionally, antibodies can cause virus particles to aggregate, further hindering their ability to attach to host cell receptors. Antibodies can also directly block the binding of viral spike proteins to host cell receptors through steric hindrance, a physical barrier that prevents the virus from connecting to the cell. Similarly, these antibodies can inhibit the fusion of viral and host cell membranes, which is essential for viral entry into the cell, also through steric hindrance. During the infection process, neutralizing antibodies can prevent essential conformational changes in the spike proteins necessary for the virus to enter host cells. For viruses that manage to enter endosomes, antibodies block their progression into the cytoplasm by inhibiting endosomal cleavage or the binding of viral components to endosomal receptors. Lastly, although not a direct neutralization action, antibodies can prevent the egress of viruses from infected cells by causing the aggregation of progeny virions at the cell surface. This mechanism is particularly evident in assays that observe multiple rounds of infection but might not be apparent in single-round neutralization tests. Neutralizing antibodies can be categorized into 4 types based on their targets and mechanisms of action. The first type includes virus-specific neutralizing antibodies. When the body is infected by a virus, it responds by producing neutralizing antibodies to clear the infection. Familiar examples of these include influenza-specific antibodies, HIV-specific antibodies, and SARS-CoV-2 antibodies. Influenza-specific antibodies target hemagglutinin and neuraminidase on the influenza virus, blocking entry and release from host cells. HIV-specific antibodies bind to the GP120 or GP41 envelope proteins, preventing the virus from attaching to the CD4 receptor and coreceptors on T cells. SARS-CoV-2 antibodies focus on the spike protein, particularly the receptor binding domain that connects to the ACE2 receptor on human cells, thereby blocking viral entry. The second type consists of toxin-neutralizing antibodies, which neutralize the effects of bacterial toxins and prevent toxin-mediated damage, such as tetanus or diphtheria antitoxins. The third type is therapeutic neutralizing antibodies, further divided into monoclonal and polyclonal antibodies. Each type of neutralizing antibody has a specific target and a unique mechanism by which it disrupts the pathogen's life cycle, making them invaluable tools in treating and preventing infectious diseases. HIV-neutralizing antibodies play a crucial role in combating HIV by targeting its envelope glycoproteins, GP120 and GP41, which are vital for the virus's ability to connect and fuse with host cells. These antibodies specifically interact with GP120 to block its binding with the CD4 receptor on T cells, a key step needed for the virus to latch onto host cells. Additionally, they target GP41 to inhibit the fusion process between the viral and host cell membranes. This dual action prevents HIV from infecting new cells, making these antibodies essential for immune defense against AIDS. HIV neutralizing antibodies have great potential in preventive strategies. Research has shown that these antibodies can be used prophylactically to prevent HIV from establishing infection in the body. This approach could be particularly beneficial in high-risk populations and is a key focus in the development of an effective HIV vaccine. And in therapeutic contexts, HIV-neutralizing antibodies can be administered to individuals already infected with HIV to help control viral replication and reduce viral load. This can potentially delay the progression of AIDS and improve the quality of life for patients. SARS-CoV-2 neutralizing antibodies specifically target the spike protein of the coronavirus, crucial for its ability to infect host cells. These antibodies bind to the receptor binding domain, RBD, of the spike protein, which is responsible for attaching the virus to the ACE2 receptor on human cells. By binding to this domain, neutralizing antibodies prevent the virus from attaching and entering cells, effectively blocking infection at its entry point. The clinical implications and therapeutic potential of SARS-CoV-2 neutralizing antibodies are substantial. Clinically, these antibodies can be administered as monoclonal preparations to at-risk individuals, potentially averting infection after exposure or mitigating severe outcomes in patients with early symptoms. Additionally, their use in immune-compromised individuals who may not respond well to vaccines underscores their therapeutic value. Developing neutralizing antibodies is riddled with challenges that are critical to their success and broad application. First, the rapid mutation of viruses leads to changes in the epitopes that antibodies target, which can decrease the effectiveness of existing treatments. This demands constant innovation in antibody design to keep up with these mutations. Additionally, these antibodies must be highly specific to their target antigens to avoid unwanted cross-reactions, which could lead to side effects. Moreover, scaling up production to meet clinical demand involves significant logistical and technical hurdles, including maintaining batch consistency and achieving high yields. Lastly, the high costs associated with developing and producing monoclonal antibodies pose a significant barrier to making these therapies accessible and affordable worldwide. These factors together underscore the complexities involved in bringing neutralizing antibodies from the lab to the clinic. Creative Biolabs offers specialized services including virus-neutralizing antibody development and virology testing, designed to support advanced research in virology and immunology. Our product line features essential tools such as virus antibodies, secondary antibodies, isotype controls, and virus detection probes for accurate diagnostics and research. Additionally, they provide virus vaccine serum and pseudoviruses, critical for vaccine development and safety testing. This comprehensive offering makes Creative Biolabs a key partner in developing effective viral therapeutics and preventive measures.